All right, so today we're going to talk about DNA and RNA. So, DNA, why do we care? What is DNA? Genes. It's what makes us us. It's what makes us us. Okay? So DNA has to be able to do two things. Store hereditary information in some sort of code. And we talked about this way back when we talked about what? What's one of the first things we talked about? Okay, organic molecules, biomolecules. So we talked about DNA and RNA back then. They are what? Macromolecule. Nucleic acid. Okay. The second thing DNA has to be able to do is be easily and accurately copied, as a copy is passed on every time that a cell divides. We talked about this just recently when we went over the what?
Cell cycle.
Cell cycle. Okay. So when is DNA copied in the cell cycle? In the S phase of interphase, right? Okay. So what properties of DNA allow it to do so? The unique structure of DNA is what allows it to be able to be the hereditary molecule that it is, and it allows it to store instructions for directing cell activity. Okay. So DNA is found where? We're going to talk about this. Where do we find it in a cell? In the nucleus. And we have talked about DNA prior. Does DNA ever leave the nucleus? No, it's too big, right? So we're going to get into a little bit of that today, but we're going to get more into that when we talk about transcription and translation of protein synthesis. So DNA stands for deoxyribonucleic acid. It is what contains the direction for making all proteins in our bodies. So DNA, because it's too big to leave the nucleus, it transcribes into RNA, and RNA is what leaves the nucleus because it's small enough to fit through the nuclear pores. RNA takes DNA's message to the ribosome, and ribosomes are responsible for making what? Protein. Okay? So that takes us back to cells. So I told you, every time we talk about stuff in bio, it all links together. Okay? And the reason that this is so important is because proteins control all of our cellular processes that take place within the body. Okay? Up here to the right, you see a diagram. This is a diagram of protein synthesis. It goes through both transcription and translation. We are going to become very familiar with this diagram as we go through each topic in this unit. Okay? So where is DNA? We already said we find it in the nucleus. But we find it in the nucleus of what type of cell?
No, the eukaryote.
The eukaryote. Yeah. Because eukaryotes have a nucleus. But we still find DNA in a prokaryote. It's just that it's not found within a nucleus. Okay? It's just in there, jumbled up. Remember I told you it looks like a bunch of spaghetti. So this takes us back that all living things contain DNA in order to carry genes, and it helps to control everything for the cell because the DNA is found in the nucleus, which is the control center for the cell. So when we talk about eukaryotic cells, specifically we talk about plants and animals. Okay? We also talk about mitosis, and during mitosis, DNA is found coiled in a chromosome. Okay? Those chromosomes take what kind of structure? They look like a what? What do chromosomes look like? X, that's it, X, okay? And then when we talk about mitosis, you already told me this today, that during interphase is when it is replicated, okay? Specifically the S phase. And that is super important, and we'll talk more about replication tomorrow. So DNA looks like a twisted ladder. So if you were to take a flexible ladder and twist it, okay, that is what DNA looks like. It is actually described as a double helix. And the reason we call it a double helix, helix for the twisty part, and double because it's made up of 2 strands. Okay? So over here is what DNA looks like. So DNA is made up of a bunch of smaller units, subunits, building blocks, monomers, and what are those monomers called? We talked about this when we did macromolecules. What's the building block of a nucleic acid? We talked about these. What is it called? A nucleotide. Okay? So nucleic acids are made up of nucleotides. So when we're talking specifically about DNA, okay, nucleotides are made up of 3 things. But specifically for DNA, a nucleotide consists of deoxyribose, which is its 5-carbon sugar, a phosphate group, and a nitrogenous base. And the bases that we find specifically in DNA are A, T, C, and G. Okay? If you missed something, I'll come back. So up here is what DNA looks like untwisted. So the backbone, or if you were looking at a ladder, the sides of the ladder, what you would hold, okay, are made up of alternating sugars and phosphates. So phosphate, sugar, okay? The middle, the rungs, or the steps of the ladder, are made up of the nitrogen or nitrogenous bases. So A, T, C, and G, okay? These bases are held together by what are known as hydrogen bonds. Okay, we saw this in our vocab this week. Hydrogen bonds are very weak, and the reason that they are weak is so that they can be easily broken in order for DNA to replicate. Okay? So note up here, because we're going to get into this, when we talk about base pairs, there's what is known as a base pairing rule. So A pairs to T and C pairs to G in DNA. So the best way to remember this is apples in a tree, cars in the garage. So A, T, C, G. Another way you can look at it is at word, right? Backwards off. I don't know that I'd call that a word, but. And then C and G look similar. The C is just missing the hook. Y'all with me? So apples in the tree, cars in the garage. Okay? So let's do a quick little review. What do the letters DNA stand for? Yep. Deoxyribonucleic acid. Good. Okay. Where do we find DNA in our cells? In the nucleus. And do you think we can see it? Why? Okay, so you can't see it with the naked eye, but you can use a microscope. Okay. All right. So this takes us to our bases. A, adenine, T, thymine, C, cytosine, and G. Okay. The bases are super important because the order of the bases are what tell the cell what protein to make. So DNA is transcribed into RNA, and then RNA goes to the ribosome, and this is where we make proteins. And this will all come together once we finish transcription and translation. Okay? So, bases in DNA are going to pair in a specific way, like I just said. A to T, C to G. T to A, G to C. So DNA is considered to be complementary, which means that each strand can be used as a template for the other half when we use the base pair rules. So if you're given a strand of DNA, you have to be able to give the complementary strand. Okay? So, for example, if you are given this strand of DNA and you are asked to make the complementary strand, all you do is use the base pair rules. So T is going to go to what? A.
C. C to G.
G.
G to C. A to G. T to A. T to A. G to C. A to C.
Beautiful. That's it. So if you are given this, you need to be able to give the complementary strand. Apples in the tree, cars in the garage. T A A T. C G G C. That's it. Okay? Let's try another one. So I'm going to say the top letter. You tell me what it's going to pair with. Ready? A.
T.
A.
G.
C.
G.
T.
A.
G.
C.
C.
G.
C.
G.
T. A. Perfect. That's it. Just don't forget it. Okay? Nice job. All right. Let's think about this. What determines the message that DNA contains? You'll see. So what determines the message that DNA contains? What did we say? What do we use to determine the order? Or what's the order of what to determine a protein? The order of the what? Order of the what? What did we just talk about? The order of the what that come out of the nucleus. What part of DNA? There's three parts. What part is it? The bases, the sugar, or the phosphates that determine the order. The bases, right. Okay, so the order of the bases are how determine the protein that we make. Okay, what always bonds with each other? What base pairs?
A to T. T to A. C to G. G to C.
Perfect. So now let's do one last example. So if one half of DNA reads the following, what's going to be its complementary strand? So A,
T.
T,
A.
C,
G.
G,
C.
yep, A,
T.
T,
A.
C,
G.
perfect. You'll get some practice with this. All right, the last little piece is RNA, and we're going to get more into this on Friday when we get into transcription, but RNA is ribonucleic acid, okay? And the differences between RNA and DNA is that DNA we said is double-stranded, RNA is single-stranded. They have different sugars, just like their name says. So DNA is deoxyribose and RNA is ribose. And we know these are sugars because what? Deoxyribose, ribose. What do we know about sugars? Take us back to macromolecules. Which sugars ended? OSE. What ends in ASE? Enzymes. Okay. All right, and the last difference is that RNA has uracil instead of thymine. Okay, so DNA is ATCG. We're not going to get into this yet, but RNA is AUCG. Okay, and I'll explain the differences on Friday. All right, so the best way to remember this is that RNA loves you. And the last piece is that there are 3 types of RNA. Like I said, we're going to talk more about this on Friday. There's mRNA, which is messenger, rRNA, ribosomal, and tRNA,